Alright, this is Dr. Larson. I'm going to bring you a lecture on causation, and this lecture is one I give in my epidemiology course. And for the implementation evaluation, this causation concept or theme is important for two reasons. First is we are trying to cause, when we implement global health programs, we are trying to cause benefit to the people that receive those programs. And so understanding how causation works for health and in public health in general is important for the implementation side. Also, when we evaluate a program, we are trying to determine the cause that the program had on health. And so we'll talk about that. And so it's an important underlying concept, and hopefully it's not too much review. Maybe hopefully it is if you've had epidemiology. It's probably a little bit of review. And if not, then hopefully it's a good one. And so in the scientific method, we start out with our theories, up here, the very first, and we generate a hypothesis. Okay. And we move forward from that hypothesis, we identify our variables, we operationalize those variables, and then we conduct a study. That's kind of how we move forward on one level. On another level, we start with a research question, and then we formulate a hypothesis, try to figure out, well, what's the explanation here? Why do we think this way? Is there any theory that underlies our hypothesis? Then we move forward with the study, and at some point we have to reject or fail to reject our hypotheses. And this process is the scientific method. A few things to note. First, a single study proves nothing. And so if we see a single paper or study on everything, then on some, say tobacco smoke and lung cancer, a single study does not prove or disprove the hypothesis. Scientific knowledge is built over time with multiple studies, and we'll talk about that in a little bit. For evaluation of global health programs or public health programs, this is a challenge because we typically only do a single evaluation. Another thing to remember is that association is not causation. And so the best example of this in my field is malaria. The word malaria comes from the Latin roots bad air. And the malaria was thought to be caused by miasma or swamp gas because it was more often there. Now, this malaria is this theory of miasma theory can keep you safe. If you avoid the swamps, then you can avoid the mosquito bites a little bit, and you can be preserved from malaria. And we can see this in the architecture of southern Italy, where the villages and hamlets are built up on these hills, and then they farm the lowlands, and so they sleep up above where there are fewer mosquitoes. Another example is with the short birth interval associated with child mortality. And so thinking about, well, does having kids close together really cause kids to die? Like a 3-year-old and a 4-year-old, does it cause them to die? Or is this just a marker for other biological processes or sociological processes in terms of access to food, access to health care, and whatnot? And so always remember that just because two things are related or observed in conjunction one with another, that does not mean that A causes B or that one causes the other. When A and B are associated, A may not necessarily cause B. You could have a reverse causation where B causes A. We could have these things together causing something completely different. Bidirectional causation means they influence each other. They could have something different causing these things. So just because A and B are associated does not mean that that causation, that A causes B. Another term that's important to know is an ecological fallacy. It's where we take population level data or population level associations, and then we make assumptions on individuals based on the population level associations. one example is that people from the South tend to have a Southern accent, and so we would commit the ecological fallacy if we found someone from the South and said, "You, personally, have a Southern accent," and in reality, the people from the cities in the South don't have much of an accent at all. So that's the ecological fallacy that is committed oftentimes in science, especially by the press. So going back to the history of epidemiology and trying to determine what causes what, Stuart Mill came out with this idea of this disease etiology, and three separate methods: method of difference, method of agreement, and the method of concomitant variation. The method of difference, basically you have different conditions, and those different conditions lead to different results, and so those conditions can be implicated as causes. For example, frostbite in the cold but not in the hot, not in the heat, and so cold may be implicated as a cause for frostbite. The method of agreement could be, go back to our malaria example, you have two swamps in two separate locations, the one swamp and another swamp, and these swamps basically, you have the similar health outcomes from these swamps, and so they agree with each other even though they're separate locations. And the method of concomitant variation is a dose-response relationship. The more you smoke, the sicker you get. The more you run, the healthier you get. The more you eat, the bigger you get. These kinds of things. So that was 1856. We've advanced a bit since then. The first advance comes from Koch's postulates, and Koch was a man that developed his theories right at the discovery of germ theory, and so these are very much postulates that are used for infectious diseases. The idea that if cholera is caused by a bacteria, then you have to find the bacteria in every case of cholera. If HIV is caused by a virus, then you have to find the virus in every case of HIV. The second one is that you can take that, whatever you found in that organism, you can grow it in culture. And so, like, if you find a cholera bacteria, you can grow that in on an agar plate and see it under a microscope as it grows. Then you can take that and put it in an animal and see the animal get sick, and then you can find that same pathogen inside the animal. Now these causes, or these postulates work really well for many diseases, but some they start to break down. For example, HIV, or for example the viruses. It's hard to isolate viruses, and they didn't have the technology to see them like they did bacterias. You have human-specific pathogens, and so animals, you could put it in an animal and nothing happens. So these are some of the issues. Koch's postulates they're still relevant today. If you suspect an infectious disease, we expect to be able to isolate that infectious disease. If you have strep throat, then you run a culture and you determine, alright, this person has strep throat, so let's get them the medicines for that. Almost 100 years later, we made a huge advance. In 1964, there was an advisory committee set up to the Surgeon General, and this was at the big hubbub, the big debate about whether smoking caused lung cancer. And so they came up with a few different criteria that the association between a suspected cause and effect, or in epidemiology terms, an exposure and an outcome, that association is consistent across time and across place. The strength of the association, so if it's very dramatic, then you should be able to see it. The specificity of the association, so the idea that HIV does not cause influenza. The temporal relationship, where you know that the exposure preceded the outcome. And then the coherence, or the common sense, basically. Does this suspected cause, does it jive with scientific theory, with understanding of biology? These were codified even further, expanded further, and made into the Bradford Hill criteria. I think Bradford and Hill were two doctors or something, two epidemiologists, in 1965. And you see a lot of the same ones: the strength, the consistency, the specificity, the temporality, the coherence. The biological gradient is that dose response, so they bring the biological gradient in. The analogy is where you see these same issues happening in the animal kingdom, and the plausibility is similar to the coherence. In addition to adding those three, they ranked the experimental evidence. Now, public health, global health, epidemiology is at a disadvantage to the hard sciences because we are concerned with the health of human populations, and it's unethical to experiment on human populations. It would be awful to introduce pathogens just so we could see what the cause is, what the effect is, or to subject someone to an all-sugar diet just so we could see. So they ranked the experimental evidence, the first being a randomized trial, and then a community randomized trial, prospective, retrospective cohorts, case control, cross-sectional, ecological, and case reporter series being the weakest. And you can look at these for evaluation and look at these study designs. Randomized or community randomized trial, those are the gold standards. If you have cohort designs or retrospective cohort, case controls or comparison designs, often the science will start from the bottom, where we get case reports or series. Someone finds some data and does an ecological study where they just look at aggregated population level data on exposures and outcome. Then you start to get even further into the weeds with cohorts and potentially a trial if you're trialing an intervention. So those are the criteria for causation that we use today in epidemiology. There's some information here on this slide about what each of these terms specifically means, and then I go through each one. I think this is an undergraduate class, so I believe you're probably with it enough to understand pre-post ideas, plausibility. Just bring up Occam's razor here, and think about this for evaluation. The simplest explanation is almost always correct, so we don't have to go looking far for that. The consistency, this is a plot of a meta-analysis that shows the different studies with the risk of cleft palate in the children of mothers who smoked during pregnancies compared to children whose mothers did not smoke. The consistency issue can be subject to publication bias, where interesting results, positive results, or negative, but interesting results are more likely to be published than non-interesting results or null results. And so that's one thing we have to be careful with the literature, is that, and there's some tests, one's called a funnel plot. The strength of the association, the higher the relative risk or the odds ratio, or the further away from one the relative risk or the odds ratio. The dose response, which is more exposure leads to more likely outcomes. Reversibility is an important one. The idea that if you take the exposure away, then the outcome goes away. And so smoking is a good example. You stop smoking, you actually cut quite heavily your risk of myocardial infarction in this study. All right, so that's the Bradford Hill criteria. You want to know those and understand those, and understand how those relate to evaluation and implementation. Now, those have been designed for single-caused outcomes, and I think as humanity we search for these single-cause outcomes. If I wear my seatbelt, then I won't die in a car wreck. It's fantastic. Simple solution, simple result. Human health is often not like that. And so some some folks developed the sufficient component cause model. And I'll talk a little bit about what sufficient and necessary causes are here. A sufficient cause means that the exposure could cause the outcome. And a necessary cause means that the exposure or the outcome cannot develop without the exposure. So here's an example of tuberculosis. To have tuberculosis disease in humans, a necessary cause would be the tuberculin bacteria in the humans. That, though, is not necessarily sufficient. Just because you have that bacteria doesn't mean you develop the tuberculosis disease. 1/3 of the population has the tuberculin bacteria in their system, but they never develop tuberculosis. And so you have to have these other sufficient causes to make that develop into tuberculosis. Now, with that understanding, this sufficient component cause model allows for the examination of risk factors. And so say you're looking at what caused, and we'll take it back to Raku's baby, and say you have 3 babies that died about that age, and you look at all the risk factors that caused Raku's baby, and then baby 2 and baby 3, and then you look across the 3 what risk factors are consistent. And so maybe baby 2, it was not a single mother. And maybe baby 3 did not work for the same person, the same farmer. And so you can look across those, see what's consistent, and then that gives you a sufficient component that you can then intervene on. And the sufficient component cause model and the sufficient necessary causes, those help us explain away the doubters, I guess, the people that say, oh, my uncle smoked for 50 years and he's doing great. It's like, yeah, public health, epidemiology, causes are rarely sufficient and necessary. They may be sufficient and there may be necessary pathogens, but to be both sufficient and necessary is rare. It's usually multifactorial. We talked a little bit about distal and proximal causes. This is an example of a causal web or a conceptual framework for lead poisoning. Here's another example for non-communicable diseases. And then there's some terms here: the predisposing factors, enabling factors, precipitating factors, and reinforcing factors. We can intervene on these different levels, and so primary and secondary prevention of health outcomes is, for public health, is basically when we, well, I won't go to that. Okay, yeah, we can intervene on these different levels. Here's another example of a causal web or a conceptual framework, and another one on child mortality, and yet one more on looks like nutritional health status, and it's cyclical in this one, so you can go back and forth. And here's one on diabetes. So that's causation, pretty short. Things you need to know: the Bradford Hill criteria, sufficient component cause model, necessary and sufficient causes, proximal, distal, precipitating causes. And these will then feed into how we talk about evaluation when we do. And as we talk about implementation and designing interventions, that's where we bring up the idea, like, why do we think that our intervention will cause a change for the better in the people that it will serve? So, thanks again. Thank you for